Hi everyone. In this presentation, we'll be summarizing and critiquing an article investigating short-term microglial depletion with CSF1R receptor inhibitors during the chronic phase of traumatic brain injury, hereby referred to as TBI. TBI and the associated tissue damage and neurological dysfunction can occur through both primary or mechanical and secondary or biochemical mechanisms. And recently, chronic neuroinflammation has been identified as a secondary injury mechanism for TBI. Microglia are the primary innate immune cells that are activated following brain trauma in order to protect the brain from injury. However, TBI is often associated with chronic microglial activation, which can lead to neurodegenerative processes and neurobehavioral dysfunction. As demonstrated in the figure on the slide here, where chronic microglial activation due to an inflammatory signal, so TBI, leads to a vicious cycle of neuronal damage and further microglial activation. In order to mediate the effects of extended microglial activation, recent studies have reported attempts to inhibit important molecules that are involved in microglial pathways during brain injury. For example, inhibition of NADPH oxidase, an important mechanism in microglial-mediated toxicity, resulted in suppression of this toxicity, reduced brain tissue loss, and improved long-term recovery. Also, inhibition of colony-stimulating factor 1 receptor, CSF1 receptor, expressed by microglial cells, resulted in over 95% elimination of all central nervous system microglia within 7 days of treatment. Although long-term elimination of microglia is likely to be harmful to the patient, since microglia perform critical functions in the central nervous system, previous studies have demonstrated that withdrawal of CSF1R inhibitors results in repopulation of central nervous system microglia, which has been shown to improve cognitive functioning, suggesting that the short-term inhibition of microglia might be the key for TBI treatment. This study investigates the effects of the short-term elimination of chronically activated microglia with CSF1R receptor inhibitor Plexicon 5622, hereby referred to as PLX, followed by microglia repopulation on neurodegeneration and functional recovery up to 3 months post-TBI. So this study utilized 10 to 12-week-old adult male C57BL6J mice, a general purpose strain that's used in research areas including immunology and neurobiology. They were housed under 12-hour light dark cycles with ad libitum or unrestricted access to food and water. The mice were subject to control cortical injury, which involved a 4mm craniotomy, followed by impact of the cortical surface using an injury device with a velocity of 6 meters per second and a depth of 2 millimeters. Or they were subject to sham injury, where they underwent the same procedure but without craniotomy or impact. 28 days post-injury, the animals were placed on unrestricted PLX 5622 diet, which resulted in 95% microglial depletion, or vehicle standard chow diet for 1 week. The mice were then all returned to a normal chow diet for the remainder of the study to allow for repopulation of the microglia. The mice performed various neurobehavioral testing around 9 to 12 weeks post-injury, including the BMWalk test was performed before and after injury to assess fine motor coordination, an accelerating rotor-rod test to assess gross motor function and balance, a Y-maze test was performed to test spatial working memory, novel object recognition was performed to assess non-spatial hippocampal mediated memory, and a Morse water maze test was performed to assess spatial learning and memory. So after neurobehavioral testing, the mice were anesthetized and their brains were processed for histological, molecular, and cellular analysis. So during histological analysis, lesion volume was measured, neuronal loss was analyzed, immunohistochemistry was performed to assess microglial morphology and classify the microglial phenotype as ramified or bushy. Immunofluorescence imaging was performed using antibodies to identify NOx2, which is involved in microglia-mediated neurotoxicity, CD68, which is a protein that's expressed in high levels in activated microglia and low levels in resting microglia, P2Y12, a receptor that's selectively expressed in non-activated microglia in the CNS and mediates process motility during early injury responses. IBA1, a marker of microglia activation, and GFAP, a marker for reactive astrocytes responding to central nervous system injury. So during molecular and cellular analysis, nanostring analysis was performed to profile for genes involved in neurodegeneration, and differentially expressed genes were determined between 4 group comparisons involving sham or TBI groups on either vehicle or PLX diets. Real-time PCR is performed to quantify expression of genes involved in NADPH oxidase or NOx2 production, as you can see listed on the slide, reactive astrocytes, and immune system responses. Flow cytometry was also performed on brain tissue to identify resident microglial cells and peripherally derived immune cells, determine caspase-1 activity, and determine interleukin-1 beta production. Finally, statistical analysis was performed and quantitative data was expressed as mean plus minus standard error of the mean.
To establish the efficacy of PLX treatment on eliminating microglia, flow cytometry and immunofluorescence staining were performed. Flow cytometry used CD11 and CD45 INT as markers of resident microglia. Immunofluorescence used P2Y12 to visualize microglia. When compared with control levels in vehicle-treated mice, oral PLX treatment led to almost complete microglia elimination within 7 days, as demonstrated by the loss of microglia staining by flow cytometry seen here in Figure 1C, and immunofluorescence signaling seen here in Figure 1B. Returning PLX-treated mice to normal chow resulted in microglia repopulation in the brain within 7 days, such that the numbers of repopulated microglia were not different to levels in vehicle-treated control brain, which is also seen here in Figure 1C. Because PLX treatment significantly improved beam walk performance, increased the amount of time mice spent on the rotarod, increased the amount of time spent with the novel object, significantly increased the percentage of time mice spent in the escape quadrant, and increased the percentage of mice who use a systematic search strategy versus a random looping one. This study concluded that delayed depletion of microglia with CSF1R inhibitor after traumatic brain injury improves long-term motor and cognitive function recovery. Next, because PLX treatment attenuated the expression of NADPH oxidase-related genes, such as CYP and NCF1 seen here, reduced the expression of key caspases, decreased the expression of an upstream pro-apoptotic gene, decreased the TBI-induced upregulation of NLRP3 inflammasome-related genes, such as NLRP3, CASP1, and interleukin-1B seen here, and reduce the number of NOX2 or CD68 expressing reactive microglia. This study concluded that treatment also altered cortical transcription related to neuroinflammation, oxidative stress, apoptosis, and neuroplasticity. Next, because treatment with PLX did not alter TBI-induced changes in GFAP cellular distribution or expression levels, they concluded that it did not affect post-traumatic astrocyte reactivity. Next, because treatment with PLX reduced the neuronal loss in both the cortex and the dentate gyrus at 12 weeks post-injury, increased the numbers of ramified microglia, which are shown here in this figure, and decreased the numbers of hypertrophic or bushy microglia in the injured cortex, which are also visualized here. They concluded that this treatment reduced histological markers of neurodegeneration and microglial activation. Lastly, because PLX treatment reduced both the size and the granularity in microglia, as well as microglial caspase-1 activity and interleukin-1 production, they concluded that delayed depletion of microglia with CSF1R inhibitor after traumatic brain injury reduced the inflammatory status in microglia. Based on these results, this study concluded that short-term depletion and subsequent repopulation of microglia during the chronic phase of experimental TBI reduces chronic neuroinflammation by altering microglia phenotype and reducing chronic NOX2 gene expression and NLRP3 inflammasome activation, and improves sensory motor and cognitive function and decreases neurodegeneration by eliminating the destructive neurotoxic microglial phenotype that may contribute to chronic post-traumatic neurodegeneration and its associated neurological dysfunction. These findings also provide support for the theory that the therapeutic window for targeting chronic neurodegeneration and improving functional recovery after TBI may be far longer than traditionally believed. To expand on how treatment with PLX may have mediated these neuroprotective effects, this study proposed that it likely involved a multifactorial mechanism that included less NOX2-mediated inflammation, which is associated with altered oxidative stress responses, and decreased expression of the NLRP3 inflammasome in microglia, which is associated with decreased release of pro-inflammatory interleukin-1. Based on these conclusions, the study also highlighted the therapeutic potential of microglia depletion and cited examples of its use in multiple sclerosis and Alzheimer's disease. However, the harmful effects of microglia depletion were also discussed to convey the need for further research on the mechanism by which microglia depletion confers neuroprotection. For example, in previous studies, microglia depletion has exacerbated post-ischemic inflammation and acute brain injury in stroke models, as well as brain neurotoxicity in models of Parkinson's disease and coronavirus encephalitis. Likewise, because CSF1R inhibitors, such as the PLX used in this study, can have off-target effects, these were discussed to support the need for further investigation into the use of these inhibitors in a clinical setting. One critique we have for the introduction is that the study supported the efficacy of therapeutically eliminating microglia during the chronic phase of TBI, followed by repopulation. However, the introduction did not define when the chronic phase of traumatic brain injury actually begins. Therefore, the period of time and the associated phase of tissue damage that should be targeted is ambiguous. Likewise, by omitting a case definition from their study, they limited their ability to guide further research on the topic.
So a strength in the introduction of this paper is the inclusion of evidence from previous studies on the effects of CSF1R inhibitors like Plexicon 3397 on microglial depletion and subsequently cognition and neuroinflammation. So the authors state that previous studies show that CSF1R inhibitors have eliminated more than 95% of central nervous system microglia, and that in a hippocampal injury model, PLX 3397 specifically, has improved cognitive recovery and reduced pro-inflammatory hippocampal gene expression. By including this evidence, the authors provide justification for performing a study that investigates short-term microglial inflammation with the use of a different Plexicon, 5622 in this study, and its impact on neurological dysfunction and neurodegeneration.
A weakness we identified in the methods section of this paper was that they excluded female mice from the study's sample population. Therefore, the generalizability of the results to the mice population was weakened. Furthermore, in a study where controlled cortical impact is inflicted in mice to model TBI, and then the effects of a potential therapy were explored, humans are the main population of interest. Therefore, the study design choice ultimately limits the generalizability of the study's result to women as well. This conclusion is supported by research indicating the role of sex in varying the type and degree of damage due to the immune response induced by an ischemic injury during the prolonged phase of tissue damage. The dramatic differences between the sexes in hormonal exposure, physiology, and biology have been implicated in these variations. Therefore, the exclusion of female mice in the study design decreased the external validity of these results, because the CSF1 inhibitor used in this study could have different effects on the neuroinflammation response of female mice and women.
So another weakness that we found in the methods section is the ad libitum or unrestricted feeding of the mouse models, and therefore ad lib delivery of the PLX 5622 or vehicle diet. With unrestricted access to the different diets, the mice may consume different amounts of food at different times, potentially resulting in variable and inconsistent dosing of PLX 5622. This may confound the results, as it's unclear if dosage amount or timing has an effect or influences microglial depletion, or if these considerations are accounted for. The author should have discussed if any discrepancies among the amount and timing of PLX 5622 or vehicle doses were found between the different mice models, and if these could have any possible effects on microglial depletion.
One critique we found for the results was that they included exact p-values in the results section. Therefore, the results were not left up to the audience's interpretation, and the study's integrity was strengthened. For example, to present the differences between the time spent in the escape quadrant between the mice treated with PLX and the vehicle control, exact p-values were reported for all their significant findings. Reporting exact p-values is useful because it shows the exact probability of observing results that extreme. Therefore, the way that significant findings are presented has the ability to paint a study in either an upright or deceptive light. By remaining transparent about the exact probability of obtaining the results, this study increased the credibility of its significant findings. One critique we had for the discussion section was that they included evidence that was contradictory to their results in terms of GFAP expression. Despite the fact that they found that repopulating microglia did not significantly affect the TBI-induced increase in GFAP expression or reactivity, this study also presented evidence that indicates that there is controversy surrounding the effects of microglia depletion on acute GFAP changes. By acknowledging that the results may not be indicative of the true relationship between microglia depletion and acute GFAP changes or astrocyte activation, this study supported transparency and increased its reputability. Likewise, acknowledging this controversy also supports the call for further research on the neuroprotective effects of microglia depletion.
So a weakness that we found in the discussion is that the authors mention CSF1R inhibitors, including PLX5622, might have off-target effects and deplete cell populations other than the CNS microglia, which may include other tissue-resident macrophages that also express CSF1R. However, they do not utilize this knowledge to measure levels of other cell populations, including peripheral immune cells and other non-brain tissues. By failing to analyze levels of other peripheral immune cells with CSF1R receptors, the authors are unable to determine possible systemic adverse effects of PLX5622 use, creating a large gap in knowledge on the safety and efficacy of this potential therapeutic. Since the authors had knowledge of these possible off-target effects from prior work, we think it would have been beneficial to include a measure for this in the methods section and would have improved the credibility of their findings. Thank you for listening to our presentation.